be using a developmental network to figure out if our dragon species will have spikes and where those spikes will appear on its body. According to a study from Philip Benfey, developmental networks are sequential activity of transcription factors that activate or repress the expression of other transcription factors responsible for developmental regulation. We will be testing various strains of the given pathway on the next slide to see which allow for skin cells to spike and where. Here are the transcription factors we were given for the dragon population. What you see here is the transcription factors used for wild type, and we will understand the reasoning further in the next slide. As you can see here, there is now a pathway using arrows and stopping symbols. This is our developmental network as it occurred in nature. The S stimulated transcription of P, but the P prevented transcription of I. The I prevents transcription of K, but the K stimulates transcription of E. E is the determinant of spikage. Spike cells are indeed occurring here. What would happen if there was a mutation that removed the P? If P is removed, the I will not be able to be stopped from transcribing. In turn, P will stop K as next in line, which means E will never be transcribed and spikes will not be made. What would happen if there was a mutation that removed the I? If the I is removed, the K will still be transcribed because nothing would be there to stop it. This means the E can produce spikes as usual. What would happen if there was a mutation that removed the I and E? Once again, if I is removed, the K will still be transcribed because nothing would be there to stop it. This is not as crucial as the E. If E is removed, there will be nothing to produce spikes, hence none will be made in this instance. What would happen if there was a mutation that removed the S and the K? If S is removed, P would not be able to be stimulated for transcription. Still, K is more crucial as it helps the start of transcription for E. Since K is gone, nothing will be there to help E, and therefore no spike cells will be made in this instance. Let's have a quick wrap-up of today's discussion on developmental networks. We learned that only the wild type and the I removal will still produce spikes. It is important to note that the wild type had no mutations to begin with, unlike the rest. So why do we see spikes on some dragon cells, but not others if their genome sequence is the same? Mutations may arise in this developmental network when the need for spike cells in an area occurs. These factors in a pathway determine how those cells will be expressed for that gene. Thanks for watching, and stay spiky, dragon friends.